Carlos Bustamente. He is professor of molecular and cell biology and physics and chemistry at UC Berkeley. And Carlos is well known, very well known for basically having introduced the field of force spectroscopy to understand single molecule reactions. So he started back in New Mexico with a beautiful idea of having how to pull a DNA molecule using gravity. And then he went on developing this concept and did a groundbreaking experiment with magnetic tweezers in 92, later in 95, 96 by using optical tweezers. And from there on, using optical tweezers, he has elucidated most of the most marvelous mechanisms that we can see in the intimate behavior of biological matter. And today he will talk about one of his latest research topics, which is understanding co-translational protein folding one molecule at a time. Carlos, welcome. Thank you very much.
Thank you. Thank you, Felix. Thank you, Felix. I'm very happy to be here, and I want to thank the organizers for inviting me. I'm going to tell you about a topic that has been of great interest to my laboratory, which is to try to understand how proteins fold. And in particular, I want to talk about how they fold when they are coming out on the surface of the ribosome as they are synthesized. We call that co-translational protein folding. And to study that, we use single molecule methods, in this case, optical tweezers. And so now, proteins in the cell begin to fold right away as soon as they are starting to being synthesized in the ribosome. But much of what we know about protein folding actually has been derived from proteins that have been studied fully formed already away from the ribosome in solution, in vitro, basically. And so we want to know how does the folding of a protein change when you study it, not in vitro, but when you actually study them as they are being synthesized during translation itself. Now, we know that actually there are some effects that the ribosome exert on proteins when they are being born. For example, it is known that they tend to be destabilized somewhat when they are actually on the surface of the ribosome. And it is also known that the rate of refolding actually is affected on the surface of the ribosome. Specifically, I'd like to show you two examples. It's being shown that the midpoint of urea denaturation for a molecule, a protein like DHFR, is greatly affected when it is on the ribosome versus when it is actually studied in solution, as shown here. And in our own laboratory, we've shown that a molecule like T4 lysozyme, when it is refolding, the rate of refolding of T4 lysozyme is reduced by more than 2 orders of magnitude when it is on the surface of the ribosome versus when it is free in solution. So the question is, are there other differences besides these ones that have been established already? And one obvious difference probably has to do with the fact that when a protein folds, the protein must actually diffuse over a potential energy surface, going from the unfolded state ultimately to try to reach the native state, which corresponds to the minimum of the energy landscape. However, in a protein that is already formed, that potential energy surface pre-exists to the folded state, whereas when the protein is being synthesized, of course, the potential energy surface is being synthesized or is growing as the protein itself is formed. And so, because of this, we may expect that actually there are big differences between the folding de novo on the surface of the ribosome versus the folding in solution of the full polypeptide. So we ask, does the folding pathway of the protein change completely, or there are maybe different intermediates that are formed in the de novo case versus the fully formed protein? And so that's the question we are actually asking here. Now, one important consideration in looking at protein folding is the fact that the rate of folding is much faster usually than the rate of synthesis of the protein. Typically, protein synthesis is slow, 10 to 20 amino acids per second. Actually, more likely in the case of eukaryotic ribosomes is even slower than this. And so, whereas folding can be very fast, occurring in the microsecond regime, and most generally will take place between 1 second and 1 millisecond. And so the question is, because of that, because this difference between the rate of folding and the rate of synthesis, it's being assumed that the process of folding on the surface of the ribosome is an equilibrium process, because for every addition of the new amino acid that is given, there is enough time for the polypeptide change to equilibrate among its multiple accessible configurations. And that's what I just wrote here, okay? So, the conclusion of that is that co-translational protein folding is believed to be an equilibrium process, occurring at equilibrium because of this difference between the rate of synthesis and the rate of folding. Okay, now, because of that, because of this inference, you can conclude or you can assume that it should make no difference whether we study co-translational folding in real time as the ribosome is actually synthesizing the protein, or if we use stall equilibrated ribosome-bound nascent chains, or RNCs. So we can actually stop the chain, the ribosome, at any one of these positions, and then we can study the folding of this part that has come out already, and these are what we call stall ribosome-bound nascent chains. And there should be no difference between studying them in these equilibrated stalled chains or in real time. That's the inference. So we have a number of questions we want to address, and the first one is, how does the ribosome affect the folding of proteins, particularly multi-domain proteins? Does it change the folding pathway? Does it change the intermediates? Does the ribosome protect the cell from the attainment of misfolded states? That's another question we want to address. And finally, is the folding pathway or kinetics different between equilibrated stall complexes that I just showed you and during active translation, where the ribosome is really translating in real time? In other words, is co-translational folding an equilibrium process, as has been assumed? In order to answer these questions, we decided to study a particular protein called calerithrine. It's a calcium binding protein, 177 residues long. Is made out of 4 EF hands. The EF hand is a helix, loop helix, and each EF hand, except for the EF hand number 2, binds a calcium ion. So, in fact, EF hand 1 and 2 form the N domain of the protein, which has 90 amino acids, and EF hand 3 and 4 form the C domain of the protein that is 87 amino acids long. Now, we are going to study this protein and try to study it and compare it, the folding in solution versus the folding on the surface of the ribosome. So let's first look at this protein as it actually unfolds and refolds in solution, fully formed. In order to do that, we are going to actually use optical tweezers, and we are going to essentially tether the molecule, the protein molecule, using DNA handles and attach them to beads, which are trapped in optical traps, as shown here. And we can actually move one of the traps relative to the other and exert force on the protein and induce unfolding of the protein. And we can relax the protein by actually moving the trap back to its original position and encourage the protein to actually reform. And this is what we observe in this case. Okay, here. Okay, this is the kind of force extension curve that you observe. This monatomic change is due to the handles of DNA, but you see that at a particular force, there is this unfolding event in which the molecule gets much longer due to the unfolding. And then if we repeat the experiment, we see that we see the same thing all over again. Now notice that in this region, the region that actually I'm indicating here with this circle, the protein is bistable. Namely, at this particular force, the protein is unfolding and it can refold. And you can see that the protein unfolds in one step, but refolds in two steps. So it actually populates one intermediate here that we can actually study. And in order to follow in more detail how this intermediate behaves, we do what we call a passive experiment, a passive mode experiment. In a passive mode experiment, we bring the protein to the force in which we see this bistability, and then we keep that distance between the traps constant. And what will happen is that every once in a while, the protein will unfold, and the bead will go back to the center of the trap, in this case, right? And so the force will drop. Whereas when the protein decides to fold, the bead will be removed from the center of the trap, and the force will tend to increase. And so if we actually look at the force with the optical tweezers, what we'll see is that we will see a type of telegraph signature that is shown here, where the protein actually is going from the unfolded state, where the force is low, to an intermediate, which is the intermediate we detected originally, which is with intermediate force, to finally eventually go to the folded state at high force. Now, among other things, notice that in this particular type of experiment, we can determine whether this intermediate is an on-pathway intermediate or not. Notice that from the intermediate, the molecule goes directly to the fully folded state. So we know this intermediate is an on-pathway intermediate. And we can actually use specifically a hidden Markov model to actually look at the transition between the unfolded state and the intermediate. In this way, we can determine the lifetime of the intermediate and the lifetime of the unfolded state, right? And the lifetime, of course, gives us the inverse of the rate constant of going from the unfolded from the intermediate to the unfolded state, or from the unfolded state to the intermediate. Okay, so in order to determine what is the intermediate in this case, we are going to compare the full protein, as shown here. You can see all four colors here, in passive mode, as shown here, with just the N-terminus of the protein that we actually have synthesized, and the C-terminus. And as I told you that actually we can determine the rate constants associated with the refolding or unfolding for each one of these processes, we expect those rate constants to be to have a functional dependence like Arrhenius-like, shown here, where the unfolding will be exponential to the force and the refolding will be exponential to the minus force times delta x. Okay, so if you plot the logarithm of the rate constants obtained from this type of passive mode experiment, you would obtain this. You can obtain that the rate of unfolding for the full protein in black increases with force, the rate of refolding decreases with force, same thing for the C-terminus, and the same thing for the N-terminus. And you notice that the C-terminus alone actually has the same rates of unfolding and refolding than the intermediate that we observe with the full protein. So that automatically tells us that the intermediate that we observe corresponds to the folding of the C-terminus of the protein. Okay, so now we know that the intermediate is the C-terminus, which is what I show here. Sorry. Oops, sorry. All right, so now the question is, does the ribosome force, okay, so notice this, in solution, you have the N-terminus and the C-terminus. The C-terminus folds first, and then the N-terminus folds, right, in solution, just out of the ribosome. However, when the protein is being synthesized, the N-terminus comes first, and then comes the C-terminus. So the question is, does the ribosome force the protein to actually fold first on the N-terminus and then on the C-terminus due to the vectorial nature of the protein synthesis? In order to answer the question, we generated these RNCs at 3 different lengths. RNC 135 amino acid, RNC 146 amino acid 146, and 157. and you can see the different parts of the protein that are out for each one, already out of the ribosome for each one of these lengths. And these correspond to refolding curves. This is not the unfolding, but the refolding curves. And notice that even though the N-terminus is completely out in the surface of the ribosome, in this case 146 and 157, there is no refolding. The ribosome is preventing the N-terminus from folding, even though it's completely out of the ribosome. It's on the surface of the ribosome, but it's out. In fact, if you compare that refolding curve to the N-terminus alone in solution, you see that the N-terminus refolds. So somehow the ribosome is preventing the N-terminus from folding when it is just appearing there. So we decided that we were going to extend a little bit longer, extend from 157 to 167, 177, and eventually 177 plus 45 additional amino acids just to remove the protein almost from the surface of the ribosome. All of this is with RNCs, with the stall complexes, mind you, here. And you see that in the case of 167, all of the N-terminus is out and part of the C-terminus is coming out. But with 177 also, you see that all the N-terminus and half of the C-terminus is completely out of the ribosome. And what we see is that with 177, you see, and also with 167, we begin to see transitions corresponding to the folding of the protein. When we look at this folding, we discover that actually it folds by 130 amino acids, which is longer than the N-terminus or the C-terminus, but shorter than the full protein. So we begin to believe that this folding that we observe here is a misfolded state of the protein. In fact, notice that when we add 45 amino acid linker at the end, the protein now doesn't form this 130 misfolded state, but first folds through the C-terminus and then through the N-terminus. In fact, let me show you in more detail the difference between the two cases. Here you see what we observe with 177. We see that the protein actually misfolds, and we can convert using the worm light chain model of polymer elasticity the forces that we observe into the number of amino acids. And you see that indeed this state, this folded state, it corresponds to 130 amino acids and is a misfolded state. However, when we have the protein completely out of the ribosome, notice that the protein now will fold through two states. One is first the C-terminus and then the N-terminus, and it will be completely natively folded. Okay, so this state is a misfolded state, and we suspected that misfolded state is due to the folding of the EF hand 1, EF hand 2, and part of all of the EF hand 3. Okay, so it's a misfolded state that doesn't occur not naturally in solution. In order to test this idea, we compare the RNC 177 on the ribosome with just the EF 1, 2, 3 in solution that we synthesize in solution. And you see that in fact, the two actually undergo the same compaction, indicating that indeed the misfolded state that we observe with the 130 amino acid corresponds to the misfolding of the EF hands 1, 2, and 3 that do not normally happen when the protein is in solution. Okay, so we may ask the question then, okay, so on the surface of the ribosome, the protein may actually misfold when it arrives to this position, but does the ribosome affect the kinetics of misfolding that we are observing? In order to answer that question, we can actually study this process on the surface of the ribosome using passive mode, and we can actually use again the logarithm of the rate constant versus force, and you can see that this is the rate of unfolding and the rate of refolding of EF13 in solution. And you see that here is the rate of unfolding and the rate of refolding of RNC177 on the surface of the ribosome. You can see that the ribosome indeed decreases the rate of unfolding, the rate of refolding by 4 orders of magnitude at 0 force, and increases the rate of unfolding by 90 times at 0 force as well. However, it turns out that the time this misfolded state remains unfolded is just less than 1 millisecond, whereas it remains misfolded by 1.5 seconds. So, what this is telling us is that the ribosome doesn't seem to really be helping too much the cell to prevent the misfolded state, because, as you see, it's 1.5 seconds lifetime misfolded and just 1 millisecond or less unfolded. And this is the conclusion we will arrive if we look at stalled RNCs. So now the obvious question is this result that we observe here, which is misfolding plus no protection on the part of the ribosome with respect to the misfolded state, which we observe with stalled RNCs, valid when the ribosome is really actively translating? So this is the question we're going to ask. In order to answer that question, we need to do this really tour de force experiment where we actually grab a ribosome that is now bound to a messenger RNA, and we are going to grab the ribosome and attach it by a DNA handle to a bead, and we are going to grab the nascent polypeptide chain to attach it to the DNA to another bead, and we are going to actually add all 20 tRNAs with amino acids, all elongation factors, EFTU, EFG, all the elongation factors in translation, in order to get the ribosome to start translating in real time. And we are going to follow at constant force, at constant distance, in passive mode, we are going to follow how the polypeptide chain is elongating. How much time do I have left? I just want to make sure that I don't run over.
Still 15 minutes.
Oh, Jesus. All right. I was very worried about actually being so. Now I can relax, right? Okay. So, Matt, let me make sure that actually I can explain you this. I get you to understand the experiment, because in this case, we need to make sure that the ribosome always starts on the same place. And then we restart the ribosome translation by adding tRNAs, all 20 tRNAs, right? And all the elongation factors. Now, in order to stall the ribosome, the messenger RNA has in front of it 3 valings, as shown here, and we don't add the tRNA for veiling. So the ribosome starts here translating, and when it gets to this point in the messenger RNA, it stops. So it always starts at the same point. Then when we add the tRNA, all 20 amino acids plus the veiling tRNA, then the ribosome will start translating the messenger RNA. And what will happen is that because we are working in passive mode, the distance between the traps is constant, and so the tension, as the polypeptide gets longer and longer, it will start to sag. And so you will see the force actually coming down, which corresponds to the synthesis of the polypeptide, okay? So this is what you are going to see here, and well, for some reason I cannot see my cursor, but if I go like this, you can see that, in fact, the force as a function of time is coming down, as you see here. This is the growth of the polypeptide in real time, and then you begin to see transitions that actually correspond to the folding of the polypeptide, as shown here, right? So, again, the force is decreasing because the polypeptide is getting longer, and then after a period of time, the protein starts actually making some transitions corresponding to its tendency towards folding or misfolding, right? Because we can use the worm-like chain model, actually, to convert the change in force to the number of amino acids that are being synthesized. We can convert this curve into that curve, and so we know how many amino acids correspond to every moment that we are actually observing, at every position in time that we are observing. And you see that at the beginning, when the amino acid 167 is arrived at here, nothing happens for a long time, and eventually something starts to happen, and you can see that that something is that the protein being, the polypeptide being unfolded, start actually visiting this state, which is the misfolded state, 130 amino acids, exactly what we observed before. If we wait a little longer, the polypeptide will continue grow by a few amino acids. And now we see that there is a competition between the C-terminus that has almost completely come out of the ribosome, which is shown here, the unfolded state, the C-terminus, and the misfolded state. So now the C-terminus is out-competing or competing with the misfolded state in the real time, right? In real-time translation. Now, what is interesting is that when the polypeptide already is 167 amino acids long, nothing happens for a while. And there's a lot of time before misfolding starts occurring, right? So what we find that actually 75% of the time, there is a lag, a period of time between arriving at 167 and the first time that we begin to observe misfolding occurring here. 25% of the time, the polypeptide has grown enough for the C-terminus to start folding, as shown here, and only 1% of the time, the ribosome actually immediately after it arrives to position 167 starts misfolding. So somehow, when the translation is occurring in real time, the polypeptide is not misfolding for a period of time. And in fact, we can look at the distribution of this time, tau, and we find that it follows roughly an exponential, and the characteristic time is 63 seconds. So the polypeptide actually can arrive at 167 and be 63 seconds on average unfolded before eventually it equilibrates and it starts misfolding. And from that point onwards, it goes unfolded, misfolded, unfolded, misfolded, unfolded, misfolded, like that. And so, interestingly, what this really means is that co-translational folding is an out-of-equilibrium process, not as was assumed, given the fact that the rate of translation is very slow relative to the rate of folding, as I said at the beginning, but actually is an out-of-equilibrium process, and the ribosome during active translation prevents the attainment of the misfolded state. Not so with the RNCs. Why? Because to generate an RNC, you have to stop the ribosome at one particular position. And it takes approximately 20 minutes for us to actually finally grab this RNC in the optical trap in order to actually study the unfolding and misfolding by applying force. And 20 minutes is a lot of time, of course, for the system to equilibrate. And we are trying to avoid, and that's why we actually observe the equilibration, which is the misfolding. So to conclude, what I want to say is that not only the ribosome does not induce an end-domain direct folding, as we may have expected given the vectorial nature of the folding process, but it suppresses folding of the end domain relative to the domain in solution, number one, destabilizes the misfolded state through both slower folding and faster unfolding, and the co-translation protein folding is an out-of-equilibrium process that can be exploited to increase the yield of the correctly folded product. Now, the 1 minute delay that we observe, 63 seconds on average, prevents misfolding and probably could, in real time in the cell, allow for chaperones to come to the rescue and prevent the misfolding, or allow for further synthesis of the polypeptide so that once the C-terminus is completely out, the C-terminus outcompetes the misfolded state and the misfolded state is prevented in real time. And finally, I want to say that equilibrium co-translational experiments that have been done by our own group, actually also, may miss key dynamics of true co-translational folding. In other words, if we want to study co-translational folding, we better study it in real time rather than with equilibrated RNCs, because they may actually be missing very important dynamics of the process. So to finish, I want to show the people that have done the work, which is Lisa Alexander, who is here. Let me just do this. Okay, so Lisa Alexander was a graduate student in the chemistry department at Berkeley, and Daniel Goldman, also a graduate student in chemistry, and Liang Minghui, who was a postdoctoral in the laboratory. And so let me finish here, and thank you for your attention.
Thank you. Thank you very much, Carlos, for this nice presentation. Actually, we have a lot of time for questions, so that's even better. So we have time for questions now. Any question you may ask? Timo?
You probably don't know the answer because you would have told us, but what is the reason for the delay then? I mean, what is the fundamental reason? Why is that long delay? Why is it so? Is there something happening? Is there protonation happening? Or is there some additional reaction happening? Can you speculate on that?
Yes, so thank you for the question. So in solution, there is no delay. Immediately it actually misfolds when it arrives to 167, with the polypeptide of 167 amino acid. On the surface of the ribosome, the polypeptide is making contact with the ribosome, and those contacts are preventing the misfolding and are holding the protein in the unfolded state, giving enough time for the rest of the polypeptide to come out so that the C-terminus outcompetes the misfolded state, and you end up with more correctly folded protein rather than the misfolded state on the surface of the ribosome. So this is, in a sense, not surprising, because in biology, as you know, these machines have evolved to do all kinds of interesting things, like including the fact that the ribosome slows down the folding of the protein that are coming out on the surface of the... And the question again is why? Why would the ribosome slow down the folding of the protein? Well, because translation is very slow relative to folding. What prevents these proteins to actually adopt a misfolded state, given that the full information that is contained in the full sequence has not come out yet? So the ribosome is holding on into the partly synthesized polypeptide and preventing it from folding, right? So this is not unexpected. What is unexpected in this case is that the behavior of the system on the surface of the ribosome, once the RNCs are equilibrated, namely after 20 minutes that we synthesize them and grab them, is very different than what we observe in real time when the polypeptide just was born. And for at least a minute, the polypeptide doesn't misfold, giving time for the rest of the polypeptide to come out and prevent the misfolded state.
More questions, comments? Jens?
Yes.
Or maybe Alex, you can go to Jens, yeah?
Thank you. Very great talk.
You must have undoubtedly considered doing the folding and refolding experiments in real cell environments with the crowding and ultimately also the chaperones. Is that something that you're considering doing?
Yeah, thank you. All of this, of course, is done in vitro with purified components. In the cell, what we expect is that other factors actually may play a role, like what you mentioned, crowding on the cell, and also the presence of chaperones that actually can affect. In reality, in biophysics, we still have actually this gap between the in vitro work and the in vivo work, and this gap still remains. The hope is that we will be able to start, and my laboratory already has published a paper along this line, actually to use purified cell extracts to do this kind of work, which is very analytical and very precise in the laboratory, but not with the usual clean buffers, but in the presence of the cell milieu, and hoping that actually we may actually find a modulation of this behavior by the presence of these other factors that may be present. Then you can, for example, immunoprecipitate certain factors from that cell extract and then see if that actually has an effect. And from the comparison between the precipitated case from the non-precipitated case, you may actually get some conclusions about what these factors may do in the cell. This is one scheme by which we may hope to actually bridge the gap between the in vitro studies and the in vivo studies. Unfortunately, that is one of the requirements. Ideally, we would actually trap the molecules inside the cell and do all of this inside the cell, but to my knowledge, that is really difficult, even if you use magnetic tweezers, as Ninke may actually mention. Even with magnetic tweezers, it may be difficult to do this type of experiments with this precision inside the cell.
Thank you.
Yes.
More questions or comments? Yeah. Francesco, is Francesco there? Oh no, there is first, probably. Yeah, maybe. I don't know who's first, but please go ahead.
Thank you for the talk. Is anything known about interaction? I mean, there must be a part of the ribosome that's doing the job of interacting with the newborn molecule. Is there anything known about this?
Yes, we've done a little bit of that, and one thing that is very clear when you look at the ribosome is the enormous amount of negative charge that the ribosome has, because it's mostly RNA, right, with some 50 proteins, but the rest is all RNA, and it has negative charge due to the RNA. And what we found in the case of T4 lysozyme, where the refolding of the protein is affected by its contact with the ribosome, is that if we increase the ionic strength, we can passivate the interaction, the electrostatic interaction between the protein and the ribosome, and that really increases the rate of refolding of T4 lysozyme. So most likely, because this effect is probably general, the kind of interaction that are involved in preventing the misfolding are non-specific with the protein. It's holding to the protein in a non-specific way through electrostatic interactions, and that allows the ribosome to do that with any protein, not just this particular protein. And we have data that suggests that that is the case, in fact.
More questions? Francesco, yeah.
Oh, thanks for the impressive experiment. I was wondering how you choose the polypeptide, let's say, to do this experiment. Is there any reason to go for that peptide, and in which extent it can be considered of general validity?
Yes.
Okay, this is an issue because, you know, proteins can be very, very different.
Right. So, yes, that's a very good question. How did we decide to actually work with this protein? We wanted a protein that was not too big, a protein that knows how to fold in solution, so that we don't have to teach it how to fold. We wanted a protein that has two domains, and so that there is a C-terminus domain and N-terminus domain, and a protein that when we study it in solution, we will actually fold through the C-terminus domain. All of which will allow us to see what the effect of the ribosome would be in the vectorial synthesis of the protein. And so, other than that, we could do this experiment with, say, T4 lysozyme, which also has two domains. It's also 168 amino acids, so it's approximately the same size. The experiment is really a hard experiment, I should say. It took us several years to actually get the ribosome to do the work that it's supposed to do, but when we wanted to do it. We, for example, you notice that we use 3 valines. In the original experiment, we put 1 valine, and the ribosome misincorporated it and continued going. So we found ribosomes that were in different positions along the messenger. So it took 3 valines to convince the ribosome to really stop there. And finally, to restart it on that when we added the tRNA valine. So the experiment is a very difficult experiment, but what I think the lesson to get out of this is that experiments as complex as this, which involved lots of tRNAs, lots of elongation factors, a machine as complex as the ribosome, is possible these days using single molecule approaches.
Okay, so we have time just one more quick question, if there is a question here.
Lovely talk. These are epic experiments. So I was just wondering, just following up on what you mentioned, that the chaperones can make a difference. Is it something that you have tried, or Is it reasonable to try? And also, you mentioned also crowding. What about
Yes.
if you induce crowding?
Sure.
What happens?
Thank you for the question. Yeah, I think that actually we haven't actually done any work with chaperones, but my ex-postdoc and professor now at Johns Hopkins, Christian Kaiser, has been actually using chaperones to actually see how the co-translational folding actually is affected. But up to this point, he has been using RNCs, stall RNCs. The lesson to learn from these experiments is that it may be worth doing the chaperone work in the presence of the real-time elongating ribosome. And finally, with respect to crowding, it's possible to, for example, add PEG and other type of crowding factors and studying them in that way, taking care of the fact that actually adding enough PEG to actually do enough crowding changes the index of refraction of the medium, and you have to recalibrate all your optical tweezers because the forces depend on the difference in the index of refraction between the bead and the medium, right? So,
Thank you.
Very quick question, Eric.
Hi, Carlos. So, I mean, in your experiment, you pull on the nascent polypeptide chain, so it's under tension, and use the worm-like chain also to get your... So it's actually not in contact with the surface of the ribosome, on average at least. So you think that it's the transient interactions that cause this delay, or what do you expect under no force?
well, no, I think that actually the force that we are applying is not enough to prevent the ribosome to actually make contact with the polypeptide. And remember, we are also applying the same force to the RNCs. And in the case of the RNCs, the misfolding occurs immediately. So this 63 seconds average time before misfolding is only occurring when the ribosome is actually translating. Even though we are applying the same type of forces when we are following in real time versus when we have allowed the system to equilibrate, obviously more than 1 minute, more than 63 seconds, and the system now has entered this misfolded state, and now it's going to misfold and unfold, misfold and unfold. So I don't think it is the tension that is preventing the interaction, because it will happen also with the RNCs, and we see that with the RNCs the protein misfolds.
Okay, so
Thanks.
thank you very much, Carlos, for this presentation, and we move on to the next speaker.